The overall goal of this procedure is to show how to determine the effects of growth factors and inhibitors in the gene expression of developing chicken embryo limbs.
This method can help answer key questions in developmental biology, such as how signaling molecules influence the growth and differentiation of cells in vivo. The main advantage of this technique is that it provides a method to test these factors directly in the embryo. Though this method can provide insights into limb development, it can also be applied to other systems such as neural tissue, somites, or gastrulation. Demonstrating the procedure will be Rabeea Mohammed, a graduate student from my laboratory.
To begin, transfer 1 milliliter of the beads from the original stock solution into a fresh tube. Allow the beads to settle, then remove the liquid and replace it with 1 milliliter of PBS. Repeat this step 5 times and store the washed beads at 4 degrees Celsius as a slurry in PBS. Select the beads for grafting by removing them from the washed stock solution using a 20 microliter pipette and place them into 1 milliliter of PBS. Using a stereoscope, choose beads that are around 100 microns in diameter. Transfer the selected beads in 2 microliters of liquid into a new petri dish containing a 20 milliliter drop of PBS. Next, remove the PBS from the beads, first with a 20 microliter pipette.